Hey everyone, welcome back to The Explainer. Today, we're diving into something that, honestly, might just be the most profound breakthrough in medicine and drug discovery we've seen in decades. A new paper just dropped detailing an incredibly powerful new AI model called MAML. And if the data holds up, MAML is poised to completely transform biosciences, exponentially accelerate drug discovery, and finally make true personalized medicine a mainstream reality. Now, it's a dense, highly technical paper, but that's exactly why we're here. We're going to break it all down so you can see exactly how this model could very soon be inventing custom cures for diseases like cancer.
Here's our roadmap for today. We're going to cover the massive drug discovery problem, biology's siloed data, how MAML connects it all, beating the specialists, a wild real-world cancer breakthrough, and finally, what this means for the future of medicine. Let's get right into it. All right, Section 1. The drug discovery problem. To really get why MAML is such a massive game changer, we first need to look at the pretty shocking reality of how we currently design life-saving medications.
Let's start with a number that is, frankly, terrifying. 90%. That is the failure rate of new drugs. From that initial eureka moment where scientists identify a promising new medicine all the way to it actually making it through clinical trials and getting approved, roughly 90% of them just flat out fail. I mean, think about how wild that is. We literally carry supercomputers in our pockets. We've sequenced the human genome. But when it comes to actually making new medicines, we're still getting it completely wrong the vast majority of the time.
And these aren't just quick, cheap failures either. This staggering 90% failure rate happens after scientists spend roughly a decade and over $1 billion trying to build something that ultimately doesn't work. $1 billion. It's like, imagine being a bridge builder, and 9 out of 10 of your bridges collapse after you've poured $1 billion into them. You'd be out of a job instantly, right? So why exactly is modern medicine hitting this massively expensive brick wall?
That brings us to Section 2, Biology's siloed data. To understand the failure, we need to take a quick step back and look at the basics of biology and how drug design is actually supposed to work in the first place. So the basic flow of life starts with your DNA, which holds your genes. Think of genes as the instruction manuals that code for proteins, and proteins are those tiny little biological machines doing all the actual heavy lifting inside your body, from building structures to controlling your immune response. But genes aren't just simple on or off switches. They have what's called gene expression. They can be dialed up, dialed down, or mutated. And subtle errors or mutations anywhere in this chain, that is what causes catastrophic diseases like cancer. A mutated gene messes up how a protein functions, and suddenly that rogue protein is telling a cell to ignore the stop signs and just grow into a tumor. So drug design usually starts by hunting down the specific part of this pathway that went wrong, the bad guy protein.
Once you pinpoint that bad guy, well, you need a weapon to fight it, and scientists generally use two main types of drugs. First, small molecules. Think of things like aspirin. They're tiny, they easily slip inside a cell, and they act kind of like a key fitting into a lock to block that rogue protein. On the other hand, you have antibodies. These are much larger proteins themselves. Think of them almost like precise biological clamps that work on the outside of cells. Now, the absolute hardest part of all this? Avoiding toxic side effects. The human body has, 1 billion moving parts. Your perfectly designed key might fit beautifully into the bad guy's lock, but what if it accidentally fits into a lock in your liver or your brain? Proving a drug stops the disease without accidentally killing the patient is by far the toughest part of the gig.
And that brings us to the core of our current crisis, what we can call the disconnected detectives problem. We have unbelievably powerful AI tools today, but disease doesn't just sit in one neat little folder. It flows from DNA to proteins to the whole body. Yet our current AI models are totally siloed. You've got Google's AlphaFold, which is mind-blowing at predicting protein structures. You've got Evo2, which is fantastic at reading DNA, and then totally separate tools strictly for screening chemicals. It's literally like trying to solve a complex crime where one detective only has the fingerprints, another just has the security footage, and a third only has the autopsy report, and they completely refuse to talk to each other. They just can't piece the clues together into one unified story.
Enter Section 3, How MAML Connects Everything. Because this is exactly where the hero of our explainer steps in. MAML is a brand new foundation model designed to look at the entire biological picture all at once.
So how does MAML actually pull this off? Well, the researchers trained it on an absolutely insane dataset, 2 billion samples across chemistry, genetics, and protein structures. But here's the catch. A small molecule string looks absolutely nothing like a gene, which looks nothing like a 3D antibody. To fix this, they engineered a modular tokenizer,
Basically, it acts as this brilliant umbrella dictionary. It takes all these drastically different raw formats, like smile strings, which are just chemical formulas written as text, or amino acid sequences for proteins, and forces them into one unified, one-dimensional text language. Once translated, they're all mixed together into a shared, multi-dimensional space. So the AI isn't just memorizing separate lists, it's actively learning the hidden, intricate relationships between a chemical, a gene, and a protein, all at the exact same time.
Moving on to Section 4, Beating the Specialists. Now, it's an awesome theory, right? But let's see what happens when this generalized biological model actually goes toe-to-toe against highly specialized AI tools in rigorous benchmarks.
Honestly, in head-to-head matchups, MAML just crushes the competition. The research team put it through 11 brutal benchmarks covering the whole drug discovery pipeline. Let's just look at the safety tests. There's a specialized model called MolFormer, which was trained strictly on over 1 billion small molecule sequences, and it was the reigning champ for predicting if a drug could safely pass the blood-brain barrier or clear FDA toxicity tests. Think of MolFormer as an elite, world-class professional swimmer. MAML, on the other hand, is a decathlete, a generalist, spread thin across chemistry, genetics, and proteins. But guess what? The decathlete outright beat the specialized swimmer in its own pool. Why? Because having a deep understanding of genes and proteins actually gives you a much better, more holistic grasp of how a chemical is going to behave inside a living human body.
But the researchers didn't stop there. They actually threw MAML into the ring with the undisputed heavyweight champ of the world, Google's Nobel Prize-winning AlphaFold 3. They challenged both models to predict if a specific antibody would successfully bind to a disease target across 7 different tests. And shockingly, MAML beat AlphaFold 3 on 5 out of those 7 targets, which raises a massive question. AlphaFold 3 can literally see in 3D, so how on earth did MAML, a model that only reads one-dimensional text sequences, manage to beat it?
The secret lies in something called intrinsically disordered regions, or IDRs. Now, textbooks usually draw proteins as these rigid, static, interlocking 3D blocks. But in reality, something like 30 to 40% of human proteins have these floppy, highly flexible sections that behave a lot more like wiggling wet spaghetti. Because AlphaFold 3 was trained on frozen, static 3D snapshots of proteins, it really struggles to predict how these floppy dynamic regions are going to move. And by the way, these regions are notorious for driving breast and stomach cancers. MAML wins because it doesn't even try to draw a static picture. It relies entirely on the underlying math and the grammar of the sequence to understand the fluid, shifting rules of the protein. It gives it a huge advantage.
Which brings us to Section 5, the cancer breakthrough. The benchmarks are cool, but the absolute most thrilling part of this paper involves a genuine, physical lab experiment testing MAML against real, human cancer cells.
The researchers needed to prove MAML wasn't just magically regurgitating its training data, so they set up a really rigorous test. They fed the AI 4 completely new drugs that were structurally alien to anything MAML had ever seen. They even proved this mathematically with a strict T-Namido similarity score, essentially a guarantee that these drugs had 0 overlap with the training data. Then, they input the genetic profiles of over 800 diverse human tumor types: lung cancer, breast cancer, colon cancer, the whole shebang. They asked MAML, predict the potency of these 4 completely unseen drugs across these 800 tumors, and then we're going to go into a real lab and physically validate your answers.
Well, MAML returned a highly confident ranking. Its #1 prediction? A drug called carfilzomib. MAML claimed this would be the absolute most potent drug against the majority of these solid tumors. And this was an incredibly bold, almost crazy prediction. Why? Because expert human oncologists firmly believed that carfilzomib, which is an already FDA-approved drug, was totally useless against solid tumors. It is currently only approved for blood cancers. Every human expert looking at this assumed MAML was about to fail miserably.
So the team goes into the lab, runs the physical experiment on real cancer cells, and the results were just stunning. To quote the paper directly, the physical lab results perfectly matched MAM's predictions, correctly identifying carfilzomib as the most potent, cascading all the way down to vudirafinib as the least potent. MAM successfully predicted the exact relative potency order across 95% of all 805 cancer cell variations. Just let that sink in for a second. An AI, just by reading text strings, figured out that a blood cancer drug would be highly effective against solid tumors, something human experts missed for decades. This completely validates a huge breakthrough in what we call drug repurposing. It means we can use AI to scan giant libraries of existing, proven safe drugs to find immediate cures for new diseases without spending $1 billion in 10 years starting from scratch.
Finally, Section 6, the future of medicine.
As we wrap up here, we really need to look at the staggering implications of an AI that actually understands the interconnected rules of biology.
Because the thing is, MAML isn't just analyzing stuff. It can actually act as an inventor. Think of antibodies like giant microscopic claws floating around in your bloodstream. The very tips of those claws, the fingers, are what physically grab onto the disease. And the absolute hardest, most chaotic, most variable finger to design is called the CDRH3 region. The researchers gave MAML a disease target and basically said, invent the amino acid sequence for these fingers from scratch so they can grab this target. MAML achieved a massive 19% improvement over the previous leading models in generating the correct sequences for this incredibly complex region. It is actively designing custom biological tools.
So, what does this all mean for the big picture? We are officially entering the MAML era. If this model holds up in the real world the way it did in these tests, the 10-year, billion-dollar gamble of drug discovery is basically over. We can instantly retest every existing drug library against every known disease. But even crazier, we can finally bring true personalized medicine into the mainstream. Imagine a doctor taking a simple sample of your blood and DNA, plugging it into MAML to see exactly where your specific genetic expression is failing, and then having the AI literally custom design an antibody from scratch to fix your unique body. Which leaves us with this final thought to chew on. MAM proves that biology isn't just a series of disconnected snapshots. It's a single, beautiful, highly interconnected language. So when AI transitions from just reading that biological language to actively inventing your personalized cares, how quickly will we eradicate our most incurable diseases? We might be a lot closer than we ever thought possible. Thanks so much for joining me on this explainer. Keep learning, and I'll catch you in the next one.